what is synthetic biology? So again, this notion of how and why. So how do we want to engineer? So we really want to think about biological engineering as a new engineering discipline. So not think about it as an ad hoc experience where you try to get this gene to work or that gene to work, but really try to understand what the design principles are that will allow you to do this efficiently, reliably, transparently, and hopefully in a safe fashion. Okay, so we're going to spend a good amount of time on understanding how to do this. But again, why is it that we want to do this, right? And so, probably not to run your next iPhone or the next video game that you have. We're not typically thinking about flasks of bacteria that can perform computation that will solve the world's problems from just a computational perspective or solve your Facebook problems or so on. But really, there are a variety of applications in health, energy, environment, security that have been described. And if you have something else in mind, certainly we would like to hear about it. Now, in terms of where this kind of field has been or is going, the notion of engineering biological systems has certainly been around for a long time. You can point at the 70s and this notion of more directed gene manipulation as a really important point, a really important transition point in our ability to genetically modify cells, genetically modify organisms. To some extent, year 2000 brought about some really interesting projects or efforts that signified to some of us in the field this notion of now starting to think about programming biological systems. So not just thinking about, for example, expressing this gene or expressing multiple genes or even inducing expression in a certain way, but really thinking about creation of new systems of interactions that allow the expression of much more sophisticated phenomena, the much more sophisticated control over what the cell is now programmed to do. Okay, and so those include, and we'll spend lots of time in understanding toggle switch. There's essentially a 1-bit memory that's been implemented in bacteria, oscillators for gene expression that goes up and down, engineered cell-cell communication. So we do think that the year 2000 was quite an important year and marked a transition in the way we think about engineering cells. Okay. Then the formation of a community started to happen afterwards. Okay, people all of a sudden realized that they're not just individuals in some lab and nobody else, everybody else thinks that they're crazy, or they don't understand why they're wasting their time making bacteria that blink with GFP or with a green fluorescent protein on and off. Okay, and so over the next several years, really the community started to form. And Synthetic Biology 1.0 was a really exciting conference that took place here at MIT that brought together many of the people that were working in synthetic biology, and also the International Genetically Engineered Machine competition that started in their iGEM. And that's still going on. And it started with 5 universities in 2004, and now it's well over 200 universities from around the world. They were able to fit it at MIT. It's no longer something that can actually be housed just at MIT. Okay, so it's really just many people from all around the world coming together and presenting their work and talking with one another. Usually undergrads, but actually we now have high school students. We have also post-undergrads that are competing in this. So it's become a lot of fun. Artemisinin production in engineered yeast was another important milestone. Then synthesis of full bacterial genome. This happened in 2008. And then afterwards, there was actually a full and functional bacterial genome that happened. And so the progress has really been, over the last few years, been increasing tremendously. Okay. Now, a really important aspect of synthetic biology and a driving force and an enabling force is DNA synthesis. Okay, and that allows us to then be able to encode complex programs. Okay? So when you think about the scales here, you often think about, for example, single genes to begin with on this type of scale, whether, usually more than 100 bases up to tens of thousands of bases sometimes. Okay? And to some extent, thinking about individual genes and expressing them came before synthetic biology. And I think synthetic biology started to formulate as a topic or as a new engineering discipline when we started thinking about genetic circuits. Okay, so going beyond the individual gene and start thinking about wiring interactions in ways that never existed before. So not even just taking a pathway from one organism and implementing a new one, but thinking about how can new activities be regulated within the cell. What does it take to actually make that happen? Okay, so we're going to spend a lot of time. We're going to spend the bulk of our time understanding that, understanding how to actually make that work. There's also notions of, can you create minimal life? Okay, what is it going to take to be able to synthesize, for example, a new chromosome, put that into some kind of chassis, and make that work? And what would be a minimal set of genes or a minimal amount of DNA that could actually support life? Okay, and then you can also think about even bigger scales than that. Can you take an existing genome and rewrite it? Okay, large scale. So can you make wholesale changes in the genome and still make it function? Okay. Now, what would be some of the different challenges at these different scales? What challenges might you encounter when you talk about genes, circuits, minimal life, genome rewrites? How might they be the same, and how might they be different?